And all dear students, as a part of health, hygiene and wellness, we are going to talk about bacteria, its definition, size and shape. I'm Vadrevu Soni from Department of Home Science. Definition of bacteria. Bacteria are tiny single cell living organisms. There are millions of different types of bacteria. Many can be found in and on your body and are beneficial to you. These bacteria make up your microbiome, which keeps your body healthy. So let's see. It is a microscopic living organism that only have one cell. The word for just one is bacterium. Bacteria are prokaryotic unicellular organisms. They are found everywhere. They are mostly abundant living organisms. They can thrive in extreme conditions like hot springs, snow, deep snows, and it is very difficult for other organisms to live. They are on your skin and in your airway as well as in mouth. They are also in your digestive system, reproductive system and urinary tract. Scientists estimate that you have 10 times more bacterial cells than human cells in your body. Coming to the difference between prokaryotic cells and eukaryotic cells. Prokaryotic cells does not have a nucleus. In eukaryotic type, it has the nucleus is present and enclosed in a nuclear envelope. In prokaryotic cells, there is no membranes bounded organelles. They are smaller in size and usually double-stranded and circular DNA is present. And typically they are unicellular such as bacteria and archaea. In eukaryotic cells, membrane-bound organelles are present and they are much larger than prokaryotic cells. They are linear double-stranded and bounded to histone proteins and can be unicellular or multicellular such as plants, animals and fungi. So this is the difference between a prokaryotic cells and eukaryotic cells in different microorganisms. Coming to the various sizes of bacteria are very small microorganisms which are visible only under a microscope, which we cannot see through a naked eye. They are having the small size in microns. Bacteria are stained by staining reagents and then visualized under high power of magnificence of compound microscope. An electron microscope is used for a clear visualization of internal structure of a bacteria. Salient features of bacteria. They are unicellular prokaryotic cells present in different shapes, sizes and arrangement. They lack cell nucleus and membrane bounded cell organelles. Bacterial DNA is found in the cytoplasm and not packed to form chromatin as in eukaryotic cells. Bacteria cell is 10 times smaller than the human cell. The outer covering of bacteria cell is the cell wall, which is rigid and provides structural integrity. The bacteria cell wall is made up of peptidoglycan or murin. Different shapes of bacteria cell are the characteristic features of the bacteria. Bacteria contains an external appendages like cilia, flagella, etc. Let's go into depth. Now first different shapes of bacteria. They again classified into spherical, rod shape, spiral bacteria and coma shape. So let's talk about the cocci, which are a single or a multiple in group of 2, 4, 8, etc. Cocci bacteria can be either round shape, oval or elongated or bean shape. These cocci are again classified as single cell cocci, which means monococcus. In pairs they are called as diplococcus. In chain they are called as streptococcus. In bunches they are called as staphylo. What is this monococcus? Monococcus is nothing but if there is a single cell cocci, then it is called as monococci. If you see a picture of here, a single cell. If it is in pair, if the two cocci, if you see in the picture, they are two together. They are called as diplococcus. In a chain, if you see the picture of your second picture, they are in chains. They are called as streptococci. And if it is our bunches, like bunch of grapes, they are called as staphylococci. So this is about the cocci. So let's know about the bacilli. In next picture, if you see, they are in rod shape. The bacilli are in rod shape. Here, this is a picture where cocci is first. These are the microscopic view. If you can see, they are cocci, bacilli, vibrio, Coccobacilli, Spirillum and Spirochetes. Now let's talk about the rod shape. Again, if they are single or in if they are single, they are called as mono. If they are in two, they are called as dibacilli. If they are in chains, they are called as streptobacilli. Or if they have both oval or spherical shape, they are called as coccobacilli. So examples you can say lactobacilli, bacillus and pseudomonas are some of the examples of bacilli. Coming to this spiral bacteria, these bacteria are spiral or helical in shape. Depending upon the thickness and the flexibility and the motility of the cell, they are again further classified into different types, two types, Spirillum and Spirochetes. The Spirillum, these are gram-negative rigid bacteria having external flagella. Now let's know what is the meaning of flagella. Flagella is is a it's a tail-like structure which helps the bacteria to move from one place to the another. It mainly helps for locomotion. So Spirillum bacteria has a facility of flagella, which means that these Spirillum bacteria can move from one place to another with the help of the flagella. Examples you can say Spirillum, Campylobacteria jejuni or Helicobacter pylori. Now what is spirochetes? These bacteria are spiral, thin and flexible. They have an internal periplasmic flagella. These are pathogenic species that can cause various or serious disease. So these are some of the picturization form where you can see a spiral form. These are all bacteria, and the second picture is about the microscopic view. Right. So these are about the spiral bacteria. Now coming to the Vibrio. These are curved and appears to be like a comma shape. They are mostly gram-negative bacteria. They are known to cause various kind of foodborne diseases. They are facultative anaerobes and have two chromosomes which replicate independently. Examples are Vibrio cholera or Vibrio parahemolysis. So this is about the Vibrio. Is a just picture. If you see the different cocci, bacilli or spiral, the first pictures are cocci where they are single monococcus. This is a double two cells together, so they're called as diplococcus. A chain form as staphylococcus, or a staphylococcus is a bunch form or tetrads, right? So they are placed in different forms. The same similar if you see a bacilli, here you can see a spiral, a comma shape or spirochete. So this is a gist of pictures of various shapes of bacteria. That's about the gram positive bacteria and gram negative bacteria. So now let's talk about gram positive bacteria. If you see the cell wall, it is a single layered smooth cell wall. The thickness of the cell wall is 20 to 80 nanometers. If you see the thick layer, also called as multi-layered because of the peptidoglycan layer, the layer for the gram positive, that is the peptidoglycan layer, is much thicker when compared to gram negative bacteria. The thiocolic acid is present in gram positive bacteria. The lipopolysaccharide is not present, and the outer membrane is also not present. Here, for gram positive bacteria, the lipid content is very low, and they are also susceptible to antibiotics. That is about the gram positive. If you see the main difference, if you can observe over here, is the peptidoglycan layer is much thicker in gram positive bacteria compared to a gram negative bacteria. Coming to the gram negative bacteria, it is a double layered wavy cell wall. The thickness of the cell wall is 8 to 10 nanometer. It is a thin layer, often single layered also. The thiocolic acids are not present, and the polysaccharides are present in gram-negative bacteria. The outer membrane is mostly present, and the lipid content is high, that is 20 to 30% more than a gram-positive bacteria, and they are very resistant to antibiotics. So this is the main difference between a gram-positive bacteria and a gram-negative bacteria. Now, let's learn about the staining procedure in the laboratory. Now, first we have to fix the cells to the slides. Later, after fixing it, now we have to add crystal violet is added over the fixed culture. After 10 to seconds, the stain is poured off and the excessive stain is removed or washed off. Later, an iodine solution is used over the smear for 10 to 60 seconds. This step is known as fixing the dye. The iodine solution is poured off and the slide is rinsed with running water. Any excess of water from the surface is shaken off. Later, a few drops of decolorized are added to the slide. Decolorizing are nothing but it can be either alcohol, ethanol, or acetone. This step is known as solvent reaction, which helps to do decolorization in gram-positive cell and stop adding decolorizer as soon as the solvent is not colored. Later, a counter stain is being added for 40 to 60 seconds and then excess of water is removed with the blotting paper or drying technique. Then the slide is then air dried and after that it is being observed under a microscope. Now let's see what is happening. From the first step, if you're adding crystal violet, what happens if it is a gram positive and what happens if it is a gram negative? In gram positive, in both the thing, because of the peptidoglycan layer, the dye is being absorbed. The dye is being absorbed and purple color is being visible. Later, when you are adding iodine, dye and moderate complexes are formed with the both gram positive and gram negative, but in gram positive, the dye adheres firmly to thick peptidoglycan layer, but in gram negative, it does not adhere to the thin layer of peptidoglycan layer. Now, adding alcohol means which it washes out the dye moderate complex because it is firmly secured in the thick peptidoglycan layer. In this negative part, gram negative part, dye and moderate complex is easily removed from the excuse me easily removed from the peptidoglycan layer with alcohol. Later, with saturated with crystal violet dye, the cell counter stains has little no effect, but colorless cells wall can easily be taken by the counter stains. Now the last step when you're adding the saffron, cell wall ranges in color from mid to purple, dark purple. As the thick layer still absorbs this, it doesn't allow the counter stain to absorb the pink color. While coming to the gram negative, because of the cell wall is very thin in nature and the upper layer of the dye, the purple dye is being removed or washed out because of the alcohol. Now it absorbs the saffron color and it turns to be a pink to red in color. So this is how you're going to identify whether the culture is either gram positive or gram negative bacteria from the laboratory procedure. Hope you understood, girls. Thank you. Thank you, girls.